In this video, we're going to continue to dive into nucleic acid structure. We're going to see how phosphodiester bonds link nucleotides in DNA and RNA. We're going to look at the DNA double helix and also some beginning structures of RNA. So just to begin, here I have a tetranucleotide of RNA. Again, we can tell RNA because at the 2' we have an OH here. And let's look at some of the features of this RNA. First, we have the bases, again, connected to the 1' position. Here we have U instead of T because it's RNA. If this was DNA, we would have a methyl here for T. So let's look at how our nucleotides are connected. They are connected by the sugar rings. So one sugar has a phosphate at the 3' position, and this is connected to the phosphate or connected to the 5' position of the next sugar ring. And this is called a phosphodiester bond. The reason for that is that you have 2 esters in a phosphate to make 1 bond. And typically, DNA and RNA is always read 5' to 3'. And what that means is you start with your nucleotide that at the 5' position, all you have is a phosphate. So it's not connected to any other sugar. We call this the 5' end. And then we can just follow along to how we name our DNA. So we start with A, then we go 5', 3', and to the 5' of another sugar we have U, so 5', 3', C, 5', 3', G. So this would be AUCG. You can create a simplified schematic of that as shown here. So here we have our bases. At the 2' position, we say if it's DNA or RNA, so this is RNA. At the 5', we say we have a phosphate. Then here we're just showing our phosphate diester bond. Then at the very end, we have our 3' end, which is just OH, just hydroxyl, so there's no phosphate here. And at the 2' again, we're saying it's RNA. And polymers of DNA and RNA are usually called oligomers. You can get more specific like I did at the beginning and say it's a tetramer because there's 4, but in general, they're going to be oligomers. RNA is typically going to be single-stranded. DNA is typically going to be double-stranded, but that's not always the case. There are cases of RNA double-stranded and DNA single-stranded. And speaking of DNA, DNA follows Chargaff's rule, saying that a DNA double strand, the amount of A's will equal the T's, and the amount of G's will equal the C's. This is not true of RNA because it's usually single-stranded, but it is true of DNA, and that's called Chargaff's rule. Now, let's talk about the 3D structure of DNA. And so 1953 was when Watson and Crick published their paper in Nature that basically described DNA's double helix. And really, their findings was based on 3 big pieces of information. One, Chargaff's rule that we just talked about. 2, they had to correctly know which tautomeric form that the bases occupy. So tautomer is when you have the same structure. So here we have thymine, but it can have different orientations of atoms. So we have the keto form and the enol form. And so back when DNA was being discovered, researchers didn't know if it was in the keto form or the enol form. Both are possible, chemically speaking. We know now, though, that DNA and RNA and the majority of the time are in this keto form. You will still find bases in this enol form, but the majority are in the keto form. So they needed to know what form the bases were in, and they also needed actual structural proof of what DNA looked like. And so here is an X-ray taken by Rosalind Franklin that shows that DNA is a double helix. To X-ray crystallographers, basically what you do is you take a crystal of your DNA and shoot it with a laser, and you just see how the laser bounces off your crystal. That tells you information about the structure inside the crystal. And so for this is a typical structure of a double helix, something you would expect if you took a crystal of a double helix. So with those three pieces of information, they could create their model of DNA. And it's the same model that we basically use now. So they were correct in their description of DNA, where you have two polynucleotide chains, and they wrap around a common axis, as shown by my laser pointer, to form a double helix. In this double helix, you have different regions. You have one region that is more wide open. We call that the major groove. And one region which is more compressed, which we call the minor groove. And it's just a repeating of this. Major groove, major groove, so on and so forth. The strands run anti-parallel. That is, they run in opposite directions. So one strand is 5' to 3' going from the bottom to the top, and the other strand is 5' to 3' going from top to bottom. But although they are anti-parallel, their handedness is the same. That is, they are both called right-handed helixes. Now, inside this DNA double helix, the bases are in the inside. And what they're doing, the bases are forming hydrogen bonds. Now, A always goes with T to form 2 hydrogen bonds, and G always goes with C to form 3 hydrogen bonds. So GC base pair is more stable than an AT base pair due to the number of hydrogen bonds. And of course, we're talking about DNA, so if this was RNA, this CH3 would be chopped off, and that would be uracil. And since each strand always hydrogen bonds with a known partner, that one explains Chargaff's rule right there, why A is always equal to T, but it also shows that each strand can be a template, and it shows you how DNA can be replicated for new organisms. Because if you have one strand based on this hydrogen bonding nature, you automatically know what the other strand is going to be. Now, in general, DNA strands are incredibly large, and so large, in fact, we talk about them in kilobases, that is, thousands of bases. And that makes sense. DNA contains the genome. And depending on the organism, the genomes will be different sizes, right? So a virus can have like a 5 kb genome. Mammals, one chromosome can be 250,000 kilobase pairs. One thing to keep in mind, though, the amount of DNA does not necessarily correlate to complexity of organism. There are plants that have much more DNA than us, but we are probably more complex. And certain organisms actually organize their DNA into chromosomes. That is just regions of organization. And chromosomes can come in different flavors based on the organisms. So if you have duplicate copies of the same chromosomes, you are called a diploid. Humans are naturally diploid. If you only have one copy, you are haploid. So our sex cells are haploid. So humans, somatic cells, 46 chromosomes, while our germline cells are haploid and have 23 chromosomes. And DNA is not just a perfect double helix throughout. Depending on the sequence, the local sequence, you can have different types of helixes, different local structures. You can even get 4 DNA strands coming together to form a quad helix. So we're not really going to go into that right now, but just be aware that DNA is just not uniform throughout the body. And RNA structure is much more complex, and that's because RNA is generally single-stranded. So here in this picture, I'm showing single-stranded DNA, and what happens is that RNA will fold on itself to form what's called a stem loop structure. That is what's being shown here. And it can form a lot of stem loops and a lot of complex 3D structure for just one stranded RNA, because you have intramolecular hydrogen bonding. So that is hydrogen bonding within the same molecule. And it's this flexibility that actually gives RNA its ability to do a lot of different things. So DNA is mostly just genetic information. But RNA can bind to organic molecules because of this complex structure. They can be catalysts. They can catalyze reactions because of this complex structure. And the versatility of RNA has led to what scientists have called RNA world theory or RNA world hypothesis. I'm not quite sure if it's a hypothesis or theory yet, but basically the idea is all life started with RNA. RNA can do everything. It can be genetic information and it can catalyze reactions. So it makes sense that RNA was the first molecule ever in all life, as we know it, sprung from RNA. And that's it for this video. I will see you in the next video.